Good morning, everyone. This is Doug McConnell speaking. I am one of the co-founders of a nonprofit called A Long Swim. We borrowed the acronym of ALS, also known as Lou Gehrig's disease, and really have tried to use a unique way to raise money for ALS research using open water and marathon distance swimming. Many of you know about A Long Swim, and so I won't go into too much of a commercial, but one of the things that we have endeavored to do in the last couple of years is tighten our linkage to the science part of the disease, rather than just raise money, really to talk about the research elements of this. Further to that very point, Dr. Hande Ozdinler of the Feinberg School of Medicine at Northwestern University in Chicago has really been the target of our fundraising and has become the scientific director of A Long Swim. Hande is going to kind of quarterback our webinar today. We have a special guest, Dr. Dr Eisen from the University of British Columbia, who's a real pioneer when it comes to brain research as it relates to ALS. I'm going to go mute here in just a minute, but for everybody who's on, as we listen to Dr. Ozdinler and Dr. Eisen's remarks, if you have questions, there is a Q&A button, I should say, at the bottom of your screen. And if we can just use that for this webinar format, put your questions in the Q&A box, and then we will kind of consolidate those questions and make sure that our panelists answer them. Anyway, without any further disruption, Hande, can I hand it over to you?
Yes, thank you so much, Doug. So today we have a very special guest. I'm honored to be in the presence of Dr. Eisen. So Dr. Eisen is Professor Emeritus from the University of British Columbia. He graduated from the University of Leeds in England in 1959. He completed his residency in neurology at the Montreal Neurological Institute, McGill University, and was appointed to the staff of Montreal Neurological Institute and University of McGill. He moved to Vancouver, British Columbia, where he developed the Neuromuscular Disease Program and the ALS Center and the clinic. So at UBC, he was Associate Dean of Research, Head of Medicine, and Head of the Neurology Department. Dr. Eisen has written extensively on ALS and edited the motor neuron volumes of both the Handbook of Clinical Neurophysiology and Clinical Neurology. He received prestigious Forbes Norris Award for compassion and love of humanity in the study, management, and support of ALS and motor neuron diseases. He's the former president of the American Association of Neuromuscular and Electrodiagnostic Medicine. And of course, Dr. Eisen has published tremendously. He published 5 books, 35 book chapters, and more than 250 peer-reviewed journals. He also received the British Columbia Community Achievement Award for his work, and he also is the recipient of the Queen Elizabeth Diamond Jubilee Medal Award for the recognition of his work in ALS. And actually, he met Queen Elizabeth also even before she was queen. So I think Dr. Eisen is a living legend, and his work in the field of ALS has taught us many important information, and I'm honored to be with him today. And thank you so much for coming and joining us, Dr. Eisen.
So, Doug and Hande, thank you so much for asking me to join in with you. You know, I'm an old guy, but I've got some interesting memories I'm going to share with you today. When I was working with the AAEM, the Electrodiagnostic Association, I used to go to Chicago every year. And I love Chicago. I haven't been for quite a while now, but I was there. It was a wonderful city. I used to look forward to going there. So I'm going to talk to you about a passion that I share with Hande. We're both very much in love with the brain in ALS, and that's what I'm going to talk to you about. By all means, interrupt me as I go along if you have questions and things you don't understand, and I'll try and answer them as we go along. So maybe if we could move to the slideshow.
Exactly.
And I guess
I don't know if we can share your screen.
Okay, I think that's
Yes.
should work now.
Yeah,
there
and
we
if
go.
we can move to the presentation
Yeah.
mode, that'd be really nice. Yes, wonderful.
Yeah, and I'm going to put on a little laser pointer. There we go. So, you know, until very recently, until the last maybe decade, ALS was always classified as a neuromuscular disease because for many years, really until the early 90s, all you could investigate was the spinal cord, the lower motor neuron through muscle. But we've come to recognize that ALS is a brain neurodegeneration, just like Parkinson's and Alzheimer's. And there are two main neurons at the beginning of ALS, the upper motor neuron and the lower motor neuron. And these two are desperately in love with each other. They're always talking and chatting with each other. But you know, like all marriages, or many marriages, one is the boss. And it turns out that the upper motor neuron is the boss. The upper neuron wears the pants and directs the lower motor neuron. Now, this was an idea, and I'll come to a little later, I developed about 30 years ago. And of course, it was controversial, and certainly not everybody agreed with me. And indeed, still some people believe that the lower motor neuron tells the upper motor neuron what to do. But I will show you arguments that make that very unlikely. So my idea is nothing is new in the world, you know. I had a friend that said, if you read enough in the library, you can get the Nobel Prize. And all sorts of stuff can be found if you dig deep enough. And the great Jean-Martin Charcot, who worked at Salpêtrière in France, formally described ALS in the 1860s. And he immediately thought the disease starts in the brain and that the problem travels down the corticospinal tracts, this long tract, and then kills the anterior horn cells. And this is what became the dying forward hypothesis. So Charcot thought of this 150 years ago. But, you know, he did this based on very few patients. He actually only saw 10 patients. So it's a mystery how he came to this conclusion, but he did. Anyway, Charcot was adamant about two things in which he was completely wrong. One is that he said ALS is never hereditary, which is obviously incorrect. But equally importantly is he said ALS never affects cognition. ALS patients never have cognitive issues. And he was completely wrong about that. And had he recognized that, in fact, cognition is an intricate part of ALS, maybe his idea with the upper motor neuron would not have died. But in fact, it then died for the next 150 or so years. And it wasn't until a very lovely lady, Betty Brownwell, who's a pathologist in London, started to investigate the pathology of the brain and ALS very deeply, raised the issue again. She didn't actually come to the idea that ALS starts in the brain, but she certainly pointed out how important the brain was in ALS. Betty actually started the Neuropathological Association in England and later spread to the rest of the world. So she was one of the very first true neuropathologists and a very, very lovely lady. Sadly, she died rather young of ovarian cancer. Anyway, after that, the story died again. And it was not until about 1988 when a very good friend and colleague of mine, Arthur Hudson, a fellow Canadian working in London, Ontario, wrote a very short letter in the Lancet and brought up the idea again that ALS is mainly derives from the upper motor neuron. And this was based on the idea that the extraocular muscles, the eye muscles, and the muscles that control the sphincters and the bladder wall usually are not affected in ALS, or at least not until very late in the disease. And that all the other muscles in the bodies have this direct connection, speedway from the brain to the spinal cord, and that something poisons the cells in the spinal cord coming from the brain. So Arthur brought up that idea. And he then tried to do an experiment where he tried to count the cells in the brain and compare that count with what's happening in the spinal cord, because he argued that if he's right, then there should be much greater loss in the spinal cord than there is in the brain. But in fact, he failed to do that. So he actually gave up on the idea and just sort of left it there. Arthur is responsible, though, for the early studies of frontotemporal dementia, of cognitive changes in ALS, that contributed very heavily in that. Now, the reason that Arthur failed in his experiments was not that they were wrong, but he didn't really understand one fundamental issue. So these upper motor neurons do two things. First of all, let me find my little pointer. One upper motor neuron can go to very many anterior horn cells in the spinal cord. And they do so not only to their own muscle, but they go to agonistic muscles and antagonistic muscles as well. And on the other hand, many of these cells in the brain, the upper motor neuron cells, go to one single anterior horn cell. And this arrangement is very important and very special and highly developed in humans. And it allows us to make smooth, very unique movements. Without this, we're jerking about all over the place when we try and move. And it allows us to make movements as gross as, say, cutting a log with a big axe to picking up a crumb from the table or a tiny piece of rice. So it's smooth development and what we call fractionated development. So in around the early 1990s, I developed this concept that ALS was in fact what I then called a phylogenetic disease of the cortical motor neuron. Now those are two big words, and let me try and explain them a little bit. So the idea of phylogeny was that it's really only humans and the neocortex which is necessary to develop ALS. And there really are no true animal models of ALS. Now, I want to be very careful about this because they're not only handy, but there are thousands of scientists throughout the world who have spent many years working on rodent models of ALS and have done amazing work. And we've learned a huge amount about the causes of cell death using rodent models. But I do want to underscore that these models are they're not models of ALS, they're models of motor neuron death. And rodents in particular do not have this very amazing cortical motor neural system. Our closest relatives, the chimpanzees and orangutans and gorillas, do, but mainly to their hand muscles. So they're very agile with their hand and arm use, much less so with their leg use. Humans have this connection to every single muscle in the body, excepting the eye muscles and the bladder muscles. So, and they're called corticomotor neurons. Let me just come to that in a minute. So the story is that these corticomotor neurons, these upper motor neuron cells, which have a highway that speeds along at great rate down to the anterior horn cells, eventually poison the anterior horn cells. And there are many mechanisms they can do this by. In this particular cartoon, I'm talking about excited toxicity, but there are many other mechanisms involved interacting together. But they excite the cell to death. It dances itself to death in this particular example. So they are the nidus, the start of what the problem is. Now, as I said, corticomotor neuron, this system of these cells that connect directly, non-stop, high speed bullet train down to the anterior horn cell in the spinal cord, allow amazingly skilled function in humans. This is why we can play musical instruments, the piano, violin, why we're able to skate, play football and dance and ballet. And then equally importantly, why we can sing opera and have amazing voice reticence when acting. And it's because of this particular corticomotor neuronal system. And there's one other thing about it. One of the main features of ALS is muscle twitching, which we call fasciculation. And in ALS, it's very diffuse and very profuse. You see fasciculation in quite a number of other diseases, but never to the extent that you see it in ALS. And it's the low threshold, those motor units which are easily excitable that are responsible for fasciculation, and they are the ones that are connected through this corticomotor neuronal system in humans. And I've tried to do some work, but not really succeeded awfully well to show that at least fasciculations are in fact driven through the brain. And I like to think of them as mini seizures, that they don't really arise in the muscle or the spinal cord, but they arise actually in the brain. And I think this is still controversial, and other people need to work on it, but I think that's going to turn out to be the case. So in this cartoon, the idea is that the strongest corticomotor neuronal input is to those muscles which we need in agility, the hand, laryngeal muscles, and the foot. And in other muscles, it's not so strong. And in the big proximal muscles, our thighs and upper limbs, there's a very weak input, and it's mainly indirect connections. And for the ocular muscles and sphincter muscles, there is no direct connection at all. Okay. And recently, Albert Ludolf in Germany and his group have actually tried to look at this clinically in patients with ALS. And indeed, they have shown that it's those muscles with the strongest corticomotor neuronal input from the brain that are weakest, both initially and are most weak in ALS. So it's the elbow flexors and the hand extensors, the thumb abductors, and knee flexors and plantar flexors. So partial foot drop is a very early feature in ALS. So it's those muscles that get the strongest corticomotor input that become weak first and become weakest in ALS. Now, I want to show you a little video that I made, which brings out the issue of what we call the split phenotype, or in this case, the split hand phenomena. So let me just try and show you that, if I can get this to work. Oh, sorry, what's happening here? I had this going before.
Maybe you can move out from the presentation mode.
Come out. No, it should. This work.
Yeah, it was working before.
Just a second. Yeah, it was.
If you can go out to the presentation and leave the presentation mode. I don't know if you can, or if you want, we can show it at the end of the presentation.
There we go. I've got
Here
it.
you go.
Yeah. So the thenar hand, which consists of the thenar muscle here and the first os interosseous, and allows us to make a pincer grip, which is a very important movement in humans and many other animals, and has a large corticomotor neuronal representation. And in ALS, this combination of muscles, it becomes wasted and weak very early in the disease, compared to the hyperthenar hand, which is relatively spared. And this is known as the split hand syndrome, is a very characteristic sign in ALS. Okay, so actually the split hand was first described by a very lovely person in the States, about 40 years ago, Asa Wilburn. People didn't take too much notice of his short paper, but then I took up this story again about 20, 30 years later, and was able to show this using rather fancy physiology. But the important point is that this part of the hand, the thenar hand here, has a huge cortical input compared to the hypothenar hand. And now there are other split phenomena described. People now talk about the split leg syndrome and the split elbow syndrome. So these all have much larger cortical representation. And it's impossible to think of how the lower motor neuron could produce this very selective loss of muscle function in ALS. Now, don't worry about this very complicated slide. This is doing some fancy physiology. What I really want to show you here is how big the cortical representation is of the thumb and the pincer grip compared to the pinky finger. And that it sort of, this is why it dies off so readily. Now, I want to digress for a moment and talk about TDP-43. And this protein has been found in about 95% of ALS patients and more than 50% of patients with frontotemporal dementia. And what happens, let me put my little pointer back a minute. There we go. So what happens is this, this protein, which is normally in the nucleus, gets pushed out and into the cytoplasm, and it begins to form aggregates, and it bungs up the works in the cell. And once you get aggregates in a cell, it can't function anymore properly, and eventually it will die. But it turns out that the aggregates probably don't form in the upper motor neuron, in the BET cells, the corticomotor neurons. The TDP-43 comes out into the cytoplasm, but it does so in a soluble form. So the story is, in this cartoon, just worry about this middle bit, don't worry about the rest. So here is the brain cell, the corticomotor neuron, and the TDP-43 here is soluble, but it's toxic. So it goes down the length of the neuron, the length of the axon that I showed you, all the way from the brain down to the anterior horn cell. And it's in the anterior horn cell where the aggregates form and start to poison, and the cell becomes dysfunctional and eventually dies. Excuse me. Of course, there are many other mechanisms of cell death. This is just one, and I don't want you to think that TDP-43 is the whole story causing ALS. Unfortunately, it's not as simple as that, but it probably is an important end product. And I think if we were to tackle this in a way, we could probably do quite a lot of good. Well, now, I had mentioned to you that Arthur Hudson, my friend in London, Ontario, had done a lot of work on frontotemporal dementia. But even back in the 80s and 90s, it really wasn't recognized very much. In fact, even reading the textbook in the mid 80s and 90s, textbook said ALS patients never had cognitive problems, which is nonsense. In fact, we now know that up to 50% of ALS patients will have cognitive issues, maybe many more depending how carefully you test them. And conversely, as many as a third of patients who initially develop frontotemporal dementia, which is the second most common dementia after Alzheimer's, will develop signs of ALS, of motor neuron disease. And both these conditions, ALS and frontotemporal dementia, share the TDP-43 story, but more importantly, one of the 30 genes in ALS we've now discovered is shared by both frontotemporal dementia and by ALS. That is the C90R F72 gene, long name, difficult. So the bottom line is that in fact, frontotemporal dementia and ALS are continuums of the same disease. One may begin before the other. And so the idea that goes back to Sherrington, that what wires together is these two, fires together, and eventually dies together. So that now we're beginning to see that ALS is much more of a brain disease than just simply the motor cortex. Well, the difficulty was that until the really early 90s, it was not possible to investigate the brain in ALS in living patients. You could do pathology, as Betty Ronwell did so beautifully, but there's always difficulties with pathology because you're looking at gravestones in many ways, and it's quite difficult to make conclusions always. So in the 90s, 3 techniques began to be developed: transcranial magnetic stimulation, MRI, which you're all familiar with, and brain network analysis. So I'm just going to say a brief word about these. I don't want to go into great detail. I was very lucky in Canada because I got, I think, the very first transcranial magnetic machine in North America. The Americans were not allowed to use it because the FDA hadn't approved it for a while. So I got a head start on using TMS in ALS and motor neuron disease and started to make some discoveries with it. And there were all sorts of different measurements and techniques you could use with it. It's really become quite commonplace now. But the gold standard is what's called threshold tracking, which measures the actual excitability of the brain itself in relation to ALS. And that was developed by Matthew Kernan's group in Australia and has become the main technique to study ALS using transcranial magnetic stimulation. You know, early on, MRI was not terribly helpful. It was rather non-specific in ALS. There were no very obvious changes that were diagnostic, and it was mainly used to rule out some other disease. Did the patient have a brain tumor or some vascular disease? But once the software developments became sophisticated, as they have now, amazingly sophisticated, the story has changed. And now with fancy MRI software techniques, one can look at the actual fiber tracks, how the cells connect with each other, and it's becoming absolutely totally amazing. But the bottom line is then that MRI studies now have shown that ALS indeed is a very widespread brain disease. And finally, and most recently, is the study of networks in ALS, and they've been studied in Alzheimer's and Parkinson's a bit more, but only rather recently in ALS, and they've shown abnormalities. There are actually increased nodes and hubs and modules in ALS as the disease progresses. And this brings me to something extremely exciting that was described in Nature just within the last 2-3 weeks. It used to be thought when the brain develops in the embryo that the neurons in the brain sort of send out sprouts and begin, you know, to connect to each other, and then later on some sort of functional capacity develops. Well, it turns out, looking at C. elegans, which is a tiny little worm with a small nervous system that you can study quite well, that isn't the case at all, that in fact functional networks that actually tell us what it is we're going to do in various aspects develop in the embryo very, very early on. And that brings me to another major issue, and that is the business of when does ALS really begin? Now, we've known for many years in Alzheimer's and Parkinson's that they begin years, if not decades, before one gets symptoms. But for some very strange reason, nobody really believed that happened in ALS. They all thought ALS begins when you become weak and your muscles waste. And I think that's nonsense. ALS also begins years and decades before you get symptoms. And that's a big problem, because once you get symptoms, it becomes very late. You've sort of fallen off the table. And so we need to find the very, very early start of ALS if we're going to really have success in stopping it, slowing it, and eventually treating it. And I don't want to sort of belabor this cartoon, but there are many things that cause ALS to progress from early on, and there is a little evidence that we've been discussing that it may even start in the perinatal period from conception, and that it's the networks that don't develop properly and lay the groundwork so that later on in life, as one ages and the aging story takes place, that neurodegeneration starts to show its ugly head. So the important thing is we have to find markers and treatments in this long preclinical and pre-symptomatic period. So let me just summarize my present thoughts of ALS. And it's a bit like COVID-19. We keep changing the story as we learn more about it. I'm sure we'll change the story of ALS as we learn more about it. But I think the origins for developing ALS begin soon after birth, in the perinatal period. And the nidus of origin lies within the motor cortices. That's the important point. And that the brain then becomes susceptible to many, many insults: inflammation, excitotoxicity, dysfunction of the mitochondria, excessive oxidation, environmental risk factors, and so forth. And epigenetics has become a very exciting and very important world in its own right. And this is important for individual sensitivity and susceptibility. And as I mentioned, environmental risk factors, and they continue to exert their influence throughout life. And eventually, in combination, these factors cause proteins, probably of many different proteins, to become dysfunctional and to aggregate and bung up the works in the cell so that they eventually die. And that neural networks, which are the beginning, rely on the difference in excitation inhibition. After years and decades of compensatory mechanisms begin to fail, and you get into the presynaptic stages of ALS. And then finally, the networks fail and ALS becomes symptomatic and progressive. So I hope that hasn't been too much of a mouthful for everybody. But anyway, putting it in a single sentence, ALS is a neurodegeneration of the brain, and that's where we need to concentrate our efforts, I think, in the future. Right, Hande.
Thank you so much, Dr. Eisen. Thank you so much. So, I did not want to disturb your presentation. The flow was excellent. And now if I may, I would like to say a couple of things about some of the comments that you made. For example, you remember you mentioned that the upper motor neurons and the corticospinal motor neurons, they are important components of ALS. And you gave the analogy of marriage, and one rules, the other one doesn't rule. Or, in my opinion, I think it is a joint effort. Like, these neurons may actually be degenerating simultaneously. I don't think that their degeneration may be really dependent on each other, even though we call these like upper motor neurons, lower motor neurons. In reality, they are born from different parents. They live in different environments. Their birth and specification are different. Their target recognition are different. They're basically very different neuron populations. And I think their survival requirements are different. Their gene expression profiles are different. So even though we call them upper motor neurons, lower motor neurons, these two are very distinct neuron populations. And I think in ALS, these neuron populations progressively degenerate. And, putting the cortex out of the picture, I don't think is a way forward. Like, we have to include the cortex in the picture, and also investigate, how much cortical dysfunction affects the function of the spinal motor neurons, because they receive, as you showed in your slides, in your clinical video, that they really have a very strong input to our muscles. And the muscles which don't have input from the cortex, they get spared in ALS, which is actually very important to reveal how the cortical dysfunction manifests clinical manifestation of ALS. So the other thing is, you remember you mentioned about, go ahead. Go.
Can I just,
of course, of course.
So I want to make sure I understand it. So you're saying you belong to the school that believes that the upper motor neuron and lower motor neuron die independently of each other and they have nothing to do with each other.
No, that is not, that I don't think I belong to that school either. I think that their degeneration is dependent on each other at a circuitry level. And because they are part of the same circuitry, and when circuitry degenerates, it's like the London Bridge is falling down, right? So it doesn't really, when the bridge falls down, it's the whole circuitry falls down. So at a circuitry level, yes, I think that their degeneration is dependent on each other, very much so. But at a molecular, genetic, and a cellular level, the reasons for their vulnerability and the reasons for their neuronal vulnerability and progressive degeneration at a cellular level may have some unique components. For example, there may be some common components, let's say mitochondrial dysfunction or protein aggregation, but there may be some very unique components that are that makes, let's say, upper motor neurons more vulnerable than the spinal motor neurons. And it could be, one could be the cortical connectivity. Let's say the cortex has this long apical dendrite and it receives input from thousands of different neurons, and they're modulated so that they can send the information to the spinal cord. But in the spinal cord, they don't have this long apical dendrite, right? They have these local circuitries, and most of the innervation is to their soma or to their local microenvironment. So even the way they are connected within their own environment is a bit different, but at a circuitry level, I think their degeneration is dependent on each other. And when upper motor neurons degenerate, I think spinal motor neurons degenerate, because spinal motor neurons receive input, a huge input from the cortex. So within that picture, I think that if we improve the health of the upper motor neurons, we will improve the health of the spinal motor neurons. There are already experiments showing that. There are already studies showing that. So that's why in our lab, as you know, we try to build therapies and even small molecules to improve the health of the upper motor neurons, because when we do so, I think that we will be able to contribute to the health of the motor neuron circuitry. So that's my thought.
No, well, so I think we agree on.
Yeah, and then the other comment that you made about the mice and rodents not represent the human connectivity, I totally agree with that, because in the human, as you know, the cortex has direct connectivity to the spinal motor neurons, right? It's a highway, as you said, direct connectivity. And unfortunately, maybe fortunately, I don't know, but during evolution, we developed this so that we will have fine skills, fine movements in our hands, in our fingers. But of course, that made us more vulnerable to spinal cord injury and to other injuries. And in mouse models, you're right, there is also this spinal interconnectivity circuit. But at a cellular level, which we study in the cortex, if you look at the neurons at a cellular level, they represent the underlying pathologies observed in humans. And I think that's why we should not really be comparing species to species. Let's say survival in mouse model did not really translate to survival in patients. So for example, as you may know, for drug discovery studies, they have looked at the survival of SOD1 model or other disease models. And if a compound was good enough to enhance their survival, the survival of the species of the mouse, then they said, okay, so the patient's survival will also increase, right? And unfortunately, that didn't translate. But we try to make the upper motor neurons fluorescent so that we can actually study them at a cellular level. And if we can improve their health at a cellular level, then maybe we can also improve the health of the upper motor neurons in patients at a cellular level. And when the cell is happy and healthy and connected within the circuitry, this may be the way moving forward for developing effective and long-term treatments. So this is the reason why we actually look at the cells, and I think you also appreciate their importance in ALS too.
Sure, absolutely.
So we have some questions. Maybe I can read to them to you. Michelle Lawrence says, how about the canine animal models of ALS? Like dogs have a mutation in the SOD1 that causes degenerative myelopathy, and the mode of inheritance is recessive. And there's incomplete penetrance, however, and assuming dogs don't have cortical motor neuron or upper motor neuron cells. So do you think dogs would be a good model to study the upper motor neurons, you think?
Well, I mean, you know, I think any animal is a model to study the brain up to extent. That's not what I'm not necessarily opposed to that. What I'm trying to stress is that there are no natural models of human neurodegeneration. You know, there are no natural models of Alzheimer's. There are no natural models of Parkinson's disease. Now, actually, having said that, I tell an untruth, because there was a captivated gorilla living in a zoo for many years that eventually ended up developing Parkinson's disease, which was confirmed pathologically. But other than that, there's been no natural models ever described in animals. So when we induce models, you know, using SOD and so on, I'm not quite sure what they are. They're certainly models of, you know, they certainly induce anterior horn cell degeneration, and in that respect are useful to study mechanisms of cell death. But I do get worried when people then equate that to human disease, because it's not the human disease. It doesn't mimic the human disease.
That's right. Yeah, it's very hard to study the you know, human disease using the other species. I think we need to really focus on the cells and mechanisms, because there are many species differences. So Ibrahim Özan asks, are there any improvements in ALS treatment? And I would say that there are many. And I think these are exciting times for ALS clinical trials and, you know, compound identification. And recently we identified NU9 together with Dr. Silverman to improve the health of diseased upper motor neurons. And hopefully after toxicology studies, we will start our clinical trials as well. And there's actually an immense development in the ALS field. So there's another question. Can you explain the toxic excitation of the horn cells by the upper motor neurons? How are the mitochondria involved in the disease? It was mentioned in one slide. So this is a question from Nicholas Gerst. So toxic excitation of the horn cells by the upper motor neurons. How are the mitochondria involved? So I can tell from our cellular assays that we find the mitochondrial dysfunction starts very early in the disease, and we see disintegration of their inner membrane. And when the mitochondrial inner membrane is disintegrated, then it's very hard for the mitochondria to produce ATP. So the other thing that happens is that the mitochondrial DNA is very has the potential to induce neuroimmune reaction. So if that DNA leaks to the cytoplasm, that would actually create multiple problems for the cell. And it may also be the site for protein aggregation to make sure that the mitochondria does not mitochondria does not leak inside the cell. So this may be one reason why mitochondrial defects become the underlying cause for upper motor neuron degeneration. And there's one more question, if I may ask. Neuropathologists have found BMAA in the brains of people with ALS, Parkinson's and ALS. So the question is, how does BMAA impact upper motor neurons? maybe you are familiar with the work of the BMA. It's the in Guanam or
Romanian ALS.
yes.
So, sorry, so I actually could spend a whole hour telling you about Romanian ALS. So I actually went out to Guam in 1990 or thereabouts to study Romanian ALS together with Donald Kahn, who looked at the Parkinsonian component, and, and John Steele, who has done a huge amount of work on Romanian disease. He actually lives out there. He's another Canadian and moved out there many years ago to work with Romanian ALS. So Romanian ALS is different. It's not the same as Western ALS, and it's actually disappeared. What you see in Guam now is Western ALS, the same as ALS as we get anywhere else in the world. And it's never really been fully worked out what caused the Romanian disease. It's gone through cycles of being genetic or toxicity from diet and so forth, or something in the water. But anyway, it's a fascinating condition, but the bottom line is I don't think we really understand what caused it. Peter Spencer in Oregon tried to poison monkeys with BMA to try to show upper motor neuron disease, and he succeeded to some extent, but again, never really fully was able to develop a model that looked like ALS. So the Romanian story is a bit like polio, which has disappeared. I mean, there was years when one could study post-polio effects, but they've almost gone. There are very few people left in the world now with post-polio syndrome.
Yeah.
And similarly, there's hardly anyone left with Romanian disease anymore, but it's a fascinating condition.
That's right. So Dr. Eisen, I would like to pick your brain on a topic. So there's also this question from Gwen Peterson. So do you think that if there were any investigational therapies, right, that target the upper motor neurons or the cortical component, and let's say we were able to improve the health of the upper motor neurons, what implications or what clinical manifestations do you think that would have on ALS patients?
Well, I think if you did that, if one could do that very early, I think you could slow the disease down and maybe even stop it. I mean, that's what I would predict or would love to see happen.
Yeah.
Because if ALS is really driven by the brain, that's what you need to be doing. It's exactly what you need to be doing.
Exactly. And I
think trying to tackle
that's right.
Go ahead.
Your colleagues in Australia, you remember you were talking about this TMS study, and then they have actually shown that this cortical hyperexcitability, which you published many years ago, like in 1980s, you have the first papers with the TMS. The cortical hyperexcitability happens very early in ALS patients. They even use it as a potential early detection marker because the cortex raises the red flag much earlier than symptom onset. And again, studies show that there is spine loss in the upper motor neurons. There's less connectivity of the cortical neurons, so that the upper motor neurons cannot really be fully modulated by other cortical cells. So if that is what's happening, then if we can improve their connectivity, if we can improve their health, and I think they would stay in the circuitry. And once they are healthy and functional within the circuitry, I would imagine that would ultimately help the health and function of the spinal motor neurons as well. So this is also the theory that we share in our lab, because I think that the solution comes from the brain, because the movement starts in the brain, right? So if the movement starts in the brain, and this is a voluntary movement disorder, so that the movement actually is impaired in patients, we really have to consider brain and the brain cells that die in ALS as a target for cellular therapy. And I think one of the discoveries that we made with Silverman about NU9, and hopefully more will follow, will show that improving the health of the upper motor neurons may have clinical implications for a broad spectrum of ALS patients, because without an upper motor neuron loss, I don't think you can clinically define the ALS patient. You have to have an upper motor neuron dysfunction, right?
Yes, absolutely, you do.
Yeah. And as you said, even Charcot, when he mentioned the disease, it was amyotrophic lateral sclerosis. So he looked at the degeneration of the corticospinal tract as one of the manifestations of the disease, right? So the cortical involvement was one of the things that made him call this another disease.
Yeah, well, his vision was that of a dying forward idea, dying forward hypothesis.
Yeah.
That's basically what he thought was going on.
Yeah.
Yeah.
All right, so let me see if there are any other questions. Dr. Eisen, how do the concepts you presented relate to the pathophysiology of ALS veterans? ALS variants such as PLS. Richard Margolin.
So that's an important question. I think with PLS, primary lateral sclerosis, it's a bit easier because that appears to be really a primary upper motor neuron disease with loss of BET cells and cortical motor neurons. So I don't have too much trouble with that. I have much more difficulty trying to explain the very small percentage of ALS patients that appear to start with pure lower motor neuron disease, spinal muscular atrophy. They're probably less than about 5 or 7%. And so that's not so easy to explain. However, people have shown for many years now that even in spinal muscular atrophy, there is pathology in the brain. So I think it probably has to do with the relationship, again, with the numbers of cortical motor neurons versus anterior horn cells. But that aspect remains to be worked out. But PLS is very much an upper motor neuron disease.
Yes. And then there's one more call. It says, for pre-symptomatic ALS.
Hande, I'm going to jump in just for a second. This is
Sure.
Doug.
Sure.
These are terrific questions, and I wanted to thank both you and Dr. Eisen for illuminating some of this. I think it was a real eye-opener for some of us to see the timeline that can go back so many years, decades, in fact, that really give us that much more momentum to solve some of these challenges, knowing that when we finally see symptoms that it could be kind of late in the game. But again, Dr. Eisen, thank you so much for joining us today. Hande, you did a wonderful job to kind of quarterback this whole thing. And just a note to the participants. Kate has recorded this webinar. It will be posted on the A-Long Swim YouTube page, and there are a couple of other surprises that are coming out that we'll also be announcing to via email and so forth. So, Hande, any parting thoughts?
Thank you so much. I mean, the only thing that I can say is thank you, and thank you, Dr. Eisen. Thank you for your time, your presentation, and for sharing your knowledge and insights with us. And I think these are exciting times for ALS research and also for upper motor neuron diseases, and understanding the importance of the upper motor neurons for many of the motor neuron diseases. So we thank you so much for your participation. And if you would like to have closing remarks, we would be very happy.
No, I thank you for giving me the opportunity of doing this, Doug and Hande. It was great fun, and I look forward to hearing a lot more from you. I'm sure you're going to do lots of exciting things in the future. So I wish you a lot of luck.
Thanks so much. Yes, sir. This is why we swim.
That's why you swim.
Thank you all.
Okay.
Bye-bye.